Welcome to the Maine Science Podcast. I'm Kate Dickerson. Today's episode is with Steve Munger, who is an assistant professor at the Jackson Laboratory. Steve is a computational biologist, and he graciously didn't object when I introduced him as a mathematician at the beginning of this episode. Steve presented at the 2017 Maine Science Festival as part of our 5-Minute Genius event. And since we weren't limited by a 5-minute countdown clock during this conversation, we were able to get into many more details about Steve's life in science and how he landed in Maine. One thing before we get into my conversation with Steve, I wanted to let you know that the MSF will be opening an online store later this month. We'll have t-shirts, some kids' clothes, and some drinkware with designs that celebrate some of the different sciences in Maine. All proceeds will help support the MSF, and you get to show off your love of Maine science. So, Steve, welcome to the Maine Science Podcast.
Thanks for having me, Kate.
I'm delighted to have you. I thought we could talk a little bit about how a mathematician ended up at a biolab. And I know you don't describe yourself as a mathmatician, but that's my entree.
I'm far from it.
Yeah, that's my entree into it. And I know your work involves a lot of visualizing of data. So we're going to have to figure out how to do this audio-wise. So that's my little teaser for people who are listening to this. I think we can do it. I think there's a lot to talk about, but
I'm still trying to figure out how to describe what I do on a daily basis.
Oh, good. So we're both in the same boat. That'll be great.
Maybe over an hour, we'll figure it out.
Okay. But I, if you could give us a little bit of background, I know like me, you did not grow up in Maine. Unlike me, you grew up in the Midwest. And so if you could kind of just tell us how you got into science and how you got to Maine, that would be really helpful.
Yeah, I think you'd be surprised how similar Michigan is to Maine. I grew up in a small town in the middle of Michigan, a farm town that could fit right in Maine. 1,000 people, pretty tight knit community, very, blue collar working community. I had great teachers growing up, but we didn't have all of the resources that some of the other schools near us had. So I wasn't able to take calculus. When you describe me as a mathematician, I really didn't have the opportunity to really pursue math in college because I was so far behind coming out of high school. And so, I ended up moving more toward the biological sciences just because the math was really difficult. I had friends that came to college with multiple AP credits in math and were able to start, in higher math. And I didn't even know what AP credits were, let alone have any. So it was a shock coming from a small town. And I bet, a lot of kids coming from schools in Maine, small schools, had that same experience. So it's just an accident that I ended up doing what I'm doing. It's just, I really shouldn't be here. We shouldn't be talking,
On the contrary, I think from what little bit I've been able to glean from your story, your happy accident is that you took some time to figure out what it is you like to do, and then you figured out how to do it.
Yeah, I just never stopped. I think often people, they end up stopping progressing and they move off to another track. And I never really had a reason to stop pursuing academic science. I came to it through a meandering path. I thought I was going to be a doctor when I went to the University of Michigan, but I didn't know any doctors growing up. I didn't know any engineers. And so I didn't really know what that entailed. And then I realized I really hated being in hospitals. And so, I graduated with a degree in biology, but I didn't know, I had never done any research. I didn't know what to do with it. And so I ended up, on a friend's couch working at a bowling alley in Michigan and being a substitute teacher at the local high school. And then I ended up, at some point after being a cook and dishwasher at a Mongolian barbecue restaurant and a few other dead ends, I had to move back home with my mom because I ran out of money. And just kind of by chance, I walked into a temp position at the Dow Chemical Company on this really cool research project. And I just fell in love with research. It had nothing to do with biological research. We were working on this really cool organic light emitting diode technology, which, most of your phones now have that technology in it. But it really, it was a small group. I had a lot of responsibilities that I probably didn't deserve at that age, but it really gave me a chance to learn about, the research process and fall in love with it. And I was lucky to meet my wife there. And then, at that point, I was starting to see I was at a disadvantage in the company. I saw the PhD level scientists as my peers. A lot of them were my friends, but I couldn't do the things they did. For me to progress in the company, I had to move over to the quality control area, which, I learned a lot about how to do reproducible research. But at the same time, I got tired of quality control and I wanted to do, research. And this was about this time that the Human Genome Project was really, on the cover of every paper. And I had remembered that I had really liked my genetics class in college. I had never done any research. I had never met the professor. I didn't meet any of my professors in college. I had no idea how to do college, how to do high school. I still have, no studying habits, anything like that. But, at the time, after I met my wife, and she encouraged me to consider graduate school, and I probably wouldn't have done it, without her pushing me, telling me that I could do it. And then it was just deciding what I wanted to do. And some of that was I didn't have the math background to really consider a hardcore, math PhD. But I was really good at puzzles. I've always been good at seeing patterns in things, patterns in behavior, patterns in data. And so, the Genome Project really seemed like the ultimate puzzle. We had all 2 billion base pairs of the human genome, but we weren't sure exactly which of those base pairs, were really important, and, which ones were genes and which ones were just kind of useless DNA. And so I was attracted to that. I didn't know what I was getting myself into. And we ended up moving 15-hour drive away down in North Carolina. Some of that was just to be on our own. We were newly married. We went down to Duke for grad school. I had an 8-week-old daughter at the time. It was naivete is kind of bliss when you're that young, because you take chances you wouldn't take, as a 43-year-old. Looking back, we were so dumb doing that.
It's amazing how much smarter you get in your 2020s, isn't it?
Oh my gosh.
and how much more you don't take those risks, right?
Yeah.
There's some value in not knowing how little you know.
Exactly. Yeah. I think you get it, the older you get, the more you get into the routine, right? You find happiness in knowing what your day is going to be like. Whereas at that point, everything was new. I met my wife and we were engaged in 3 months after we met, and then married 6 months after that. And then she was pregnant a month after. It was a whirlwind. And so I had not gone directly into grad school after undergrad. I had spent 5 years doing this meandering path before we went back. And so I was one of the older new grad students at Duke in the genetics program. And I was the only one with, a baby in tow. And most of these kids, I'd call them, but, they're my friends, but they're younger than I am. Most of them had done research. They knew exactly what they wanted to do. And I just, I had no idea. I thought I was, going to work on human genetics and, be a hardcore data analyst. But you do these lab rotations your first year in grad school, and it kind of gives you an opportunity to try out a few different things. And so my first lab rotation, I was in a human genetics lab. And so they were trying to find regions of the genome that were associated with disease susceptibility in human populations. And so, it was a lot of just looking at like one spot in the genome and trying to just see if it was correlated with, disease outcome. It was pretty boring, to be honest with you. So then I decided, well, I don't really want to do that. So I'll go the opposite. I'll go to the far other end and I'll work in a yeast lab. And so, yeast are an amazing genetic model organism. There's all kinds of things you can do with yeast. But then I found out that, yeast don't move. they don't have appendages so much. And, and at that point I was struggling with my classes too, because there's this expectation when you start grad school that most people have some lab experience working in a biological research lab. And I had worked in a applied physics lab, but I wasn't using pipettes and things like that. And there's also a way of doing biological research that, once you've worked in a lab, it comes naturally to you. But if you haven't done that and you're a first year grad student and your professors are asking questions on exams that kind of assume you have that background, it's frustrating. And it was to the point where we were considering leaving Durham and moving back to Michigan, and just calling it a day. And then I was lucky enough, to meet an amazing professor who became my PhD mentor. And I ended up working on mouse gonads for my PhD research, which, it boggles my mind that that's what I ended up studying, but it's an amazing organ.
So a couple of things that struck me. One, I definitely introduced you incorrectly as a mathematician, but I for two reasons. One, pure mathematicians would be horrified that I say that what you're doing is math, as my son would be happy to point out to me.
Right.
But you do really a deep dive into data analysis and in ways that I don't think people, and we can get into that, ways that I don't think people realize biologists have to do nowadays.
Yeah.
Also, I'm struck by the parallels between what you experienced in undergrad and what you experienced in your beginning of grad school, where you didn't have the background that your colleagues had or your peers, but you figured out a way to make it happen. And it sounds like maybe it was just lucky for undergrad, but for grad school, you found this person. Did you do a rotation under your PhD advisor, or was it flat?
Well, she was teaching a module in one of the grad courses. And so we had this genetics course where different professors would teach about the particular model organism they worked on. And so she worked on the mouse. So she was teaching the mouse genetics section, and she was teaching about these things called quantitative trait loci, which is just a great genetic puzzle. And I ended up doing really well on her exam. And I went to, pick up my exam results from her in her office, and we started talking. And she asked me where I was doing my third rotation. And I was honest with her. I told her we were thinking about leaving. And she said, you should consider doing a rotation in my lab. And, that one bit of outreach from her completely changed my life. I mean, it was, I fit right in with that group. I felt like I had been there forever. I got a lot of support, for the first few weeks when I really needed it. And then I was off to the races on my research. It's funny, you talk about math and data analysis, and there's really been a shift, and it didn't happen right after the genome was published, 20 years ago. And so when I started grad school in 2000, I don't even want to say.
I'm older than you, if that helps.
I think it was 2005 when I started grad school. There still was not, we were not learning bioinformatics and computational biology. There was not courses in the genetics program for that. And so when I started really being attracted to, these large data sets in what we call genomics data, I didn't have any coding background or skills. And so, I really ended up taking a postdoctoral fellowship just to learn that. I made a complete shift. I was doing developmental genetics, and then shifted, when I came up to Maine, shifted to a computational project just so I could learn. And now I look at the students coming into our graduate program, and most of them already know how to code. They've learned that in undergrad. And then they learn it more in grad school. And so it's just this expectation that you have to know how to handle big data sets. It's so ubiquitous. And if you don't know how to handle these big genomic data sets, you're really at such a disadvantage because you have to depend on other people to do that work for you. And if you have trust issues like I do, you want to be able to do all of it by yourself if you need to.
So how does that affect kids like you who went to school and didn't quite even realize what they didn't, you said you didn't know what an engineer did. I went to an engineering school. I had no idea. It wasn't until I was a sophomore at an engineering school and went, oh, that's cool. I'm not going to do that because I was already so far into my science degree. We're going far afield here, but
no, it's good.
How would you recommend that we even start figuring out how to explain that the way that people my age who are making the policy decisions are creating, they're often thinking about what it was like when they were in school as opposed to what we need now?
I mean, I think, one of the biggest challenges and having grown up on one side of the divide and now being in a place where, I have so much privilege and I see things, I see that, the rules are different for people who grow up in these well-to-do communities. And it's really, I think there are a ton of kids out there in small towns and big cities that may not, have access to these resources, but could be amazing scientists, amazing engineers if they were exposed to that opportunity. And I think really that's the challenge, is providing these opportunities for kids to learn about science who may have never met a scientist or known an engineer. There are all kinds of internship opportunities at different labs across the United States for high schoolers and for undergrads. I didn't know about any of that. No one came to tell me, hey, you should look into this research internship at the University of Michigan. I was working at a grocery store bagging groceries. I was mowing lawns. Even in undergrad, I didn't know that you were supposed to take like an internship in the summer. I worked at an amusement park, or I worked at a grocery store. I wasn't doing research. And so I think it's doing the outreach, but also providing the resources, providing a little more equity and opportunity, and making sure that we're not losing these kids in these small towns who could end up curing cancer, right?
I realize
I don't know if I answered your question. But.
No, you did. And it made me realize how incredibly self-serving it may sound to people listening to this, because that's what we're trying to do at the festival. But I'm well aware that everything that we're doing with the festival is I don't want to say it's a one-off, because I don't think of it that way, but it's such a small sliver. There's so many other ways that, we need many, many different hands and many, many different approaches. So.
Yeah, it's tough, because a lot of times you end up preaching to the choir a little bit, right? I mean, who is listening to this podcast right now? I don't know, but I would bet that it's a lot of people who are professionals, whose children are going to know what to do in college. They're going to take them on college tours. A lot of them probably support the local PBS station, and the challenge is up, to us to kind of get outside our bubble. And now that, I knew growing outside of that bubble, I know how different it is. And, and I work to get to the point where my daughter and son could be inside the bubble and have a lot of the privileges that I didn't have. But at the same time, I feel a little guilty for that, because like, they don't know what other kids have to go through. And, they've always grown up, having certain advantages. And so it's, I feel guilty sometimes for being able to do what I do. And, I get, how many people get to get paid for thinking, essentially.
Right. Well, first of all, I would say, please don't feel guilty.
Oh, well, come on. I grew up Catholic.
Okay, fair enough. I also grew up Catholic, so maybe we should leave that for a different podcast. You said your PhD work was with mouse gonads.
Yeah.
So first of all, I want to know why that's so fascinating, besides just you don't often hear that in normal conversation with people. But secondly, if you could give us that and then tell us how you got to Maine and the work you're doing now. Again,
okay.
Just run with all of that.
No, we're going to focus. I'm not a very focused person.
I will circle you back to it.
No, gonads are amazing. And I didn't appreciate it until I was 25. But it turns out that, the gonad is the only organ in the body that when you're developing as an embryo, it can become two completely different organs. And so, the gonad primordium, each cell in that gonad has to decide if it wants to become a testis. Most of the time, in XY males, it will become a testis. Or in females, XX females will become an ovary. And so it's this really unique pool of cells early in development that, why do those cells usually decide to become a testis if they have a Y chromosome? And if they don't, they become an ovary. And it turns out it's not always that case, and so genetic differences between people and genetic differences between different laboratory strains of mice can cause some to be more susceptible to having those cells, shift to the alternate sexual fate. And so it turns out there's this really complicated process that is normally just set off by this little gene on the Y chromosome called SRY. And, it's this little weak gene that it's only expressed, it only makes proteins for a very short amount of time and a very small amount of protein. And that sets off this kind of cascade that then is, it's like an avalanche. And so you then start this, upregulation of these testis networks, gene networks, and then you end up with a testis. But you can't just upregulate the genes in the testis network. At the same time, they're also downregulating the genes that would make an ovary. And so if you fail to downregulate, to repress those ovarian genes sufficiently, those cells can then sometimes will transdifferentiate into their ovarian cell type. And so there are different cell types in the gonad that each one has to decide, am I going to be a Sertoli cell or am I going to be a granulosa cell? Am I going to end up being sperm or am I going to end up being an egg? And it's really this amazing process and really helped me fall in love with genetic networks and with using the genetic differences between different people and different mouse strains to understand the biology. And so, it really showed me that our genetic differences can be used as like a roadmap to understand, why we have differences in disease susceptibility. And so it's funny how I ended up in Maine. My PhD mentor was on the board of scientific counselors at the Jackson Lab. So she had a lot of connections to the Jackson Lab. My PhD research was really based on earlier research done by a professor at the Jackson Lab named Eva Eicher, who is amazing. And I've gotten a chance to know her. And she's a lot of fun. She's from Michigan. And so not long after I joined my PhD mentor's lab, so my PhD mentor is Blanche Capel. I want to make sure I say her name. She's amazing. Not long after I joined her lab, we were talking about genetics one day. And she just said, you're a real geneticist. You really think about genetics. And she said, you're going to end up going to the Jackson Lab in Maine. She said, that's where that's where they do real genetics. That's where you learn real genetics. And she was right. I ended up going to some scientific conferences, and there was this guy that was doing this amazing research, the stuff that I wanted to do, using these genomics technologies and doing genetic mapping, on these. And he's this statistical geneticist named Gary Churchill. He's at the Jackson Lab. And so when I was getting ready to graduate to defend my thesis, I emailed him in February and said, I'm interested in doing a postdoctoral fellowship with you, do some postdoctoral training. And he said, sure. He said, come out in May. So he brought me, I didn't know it at the time why he wanted to wait till May, right? He didn't want to bring me to Bar Harbor in February.
Smart man.
So I went up there in May, and it was amazing. The scientific community there, I gave a research talk on my research, and many professors showed up to that. And I was amazed by that, because normally, at student talks, a lot of times it's just fellow students and not many professors show up. But people were interested in what I had done. And it was just this real feeling of support. And then, we went out to dinner in Bar Harbor, and it was a beautiful night. And after dinner, I was walking around, down in Agamont Park, looking at the Porcupine Islands. And I called my wife and I said, I think we have to move to Maine. And she said she cried after she got off the phone. And she told her friends the next day, I think we have to move to Maine. So I convinced her to, by that point we had 2 kids, 2 cats, and we took a chance moving up to Maine. We were really nervous about moving back to a small town after living in this really diverse community, Durham in North Carolina. But it's just such a unique place. It's just such an amazing place to do science. And then you see your colleagues and your students out in the community. And, it's just this really unique setting. And I was really lucky to have the chance to stay here and start my own research program. And, it wasn't a difficult decision. After 3 years of a postdoctoral fellowship, our kids were doing really well in the school system here. We were connected into the community. It was just things I love about Maine. If we can talk about all the things I love about Maine for a second. So when we first got here, we moved here in October of 2011, end of September. We had gotten this rental house at the last minute. Someone at the lab knew someone who knew someone, whose parents had a house that they were no longer in. And so we show up at this house, and there's a giant pan of lasagna in the fridge for us, waiting for us. So our landlord was this amazing lady who was a scientist at the lab, a retired scientist at the lab. And, that just, from there on, seeing the pile of free stuff at the end of the road all the time. I mean, that's such a uniquely Maine thing. And it's an amazing thing, and to this day, if you look on your local Facebook, every locality has their barter and swap page on Facebook. We have the Bar Harbor Barter and Swap. And it's not just a place to sell things. It's a place to give to charities, to talk about like, what do people need? And it's, it's so much more than just a place to sell. It's not like Uncle Henry's, right? So it, right away, we were we loved it, and then, we went to our first chili cook-off, and, it had all the best things about small towns and not many of the bad things. Growing up where I did, sometimes, there was a lot of gossip. Everyone knew everyone's business. Mainers tend to mind their own business, which is great. I think that's just inherent to Maine. People are, they want to do things themselves. They're very self-sufficient. They will help you if you ask for help, but they will also give you space and, and mind their own business. I'm so happy that our kids are able to grow up, learning those values. It's a really great place. And so now, what am I doing now? You're looking at me over this Zoom, like, get to the point.
No, I'm having a really hard time not agreeing with just about everything you say and giving my own examples.
Well, give your own examples.
Well, so it's really interesting. I grew up in upstate New York, and my husband's an academic, so we've traveled all over the place. And the only two places where we have landed where I felt literally instantly at home, and I can't even tell you why, was Maine and Ireland. Yeah. Just the second we got to both places, I knew I could live in either place for the rest of my life and be really happy. And there's a lot of similarities between the two, even though, they are two different places separated by a common language and all of that. But one of the things I treasure most about Maine and Ireland to a certain extent too is what you said, that, there's an understanding that they're there to help you if you ask for it. So you need to ask.
Yeah, you definitely need to ask.
And some people don't know that is surprising, but there is an understanding that there's a lot of, for me, this is a little bit of diversion. There is huge diversity in both places, and it isn't necessarily with race as how most people think about it, but there is a gigantic cultural and economic diversity that is always just under the surface and is not necessarily this burden to carry. And by that, I mean, like, you're not necessarily looked down upon because you don't have all the wealth.
Absolutely.
But it's still an issue that we have to deal with. And we don't always talk about it in the way we should, which isn't at all. I think the things that Maine struggles with are struggles everywhere. It's just super nuanced, and we're willing to talk about it, but someone's got to bring it up. Yeah,
absolutely. And I think, growing up in a small, pretty poor town, and growing up well below the poverty line, I didn't know we were poor because most of my friends were under the same circumstances, right? And, I grew up in a single parent household, which didn't seem out of the ordinary to me because most of my friends' parents were divorced, and it's a completely different world where I am now. Most of my friends are in stable marriages, have that life, and it was completely different. And so I think a lot of kids are growing up in Maine in some of these small towns, and they don't really realize that they're starting, 40 yards behind other people in the 100-yard dash, right? And, a lot of kids who have these privileges don't understand they're starting on the 40-yard line. But you're right. There's a lot of economic diversity in Maine. Huge. And there's very little racial diversity overall, but boy, I mean, there's a lot of economic diversity. And that, I really think, we need more outreach to bring in kids from these, communities like the one I grew up in, bring them in to learn about research, to be exposed to people doing engineering and things like that, just to kind of, show them that this exists. I mean, just if I had known, that what biochemical engineers or, what engineers did, if I had known an engineer, I bet I probably would have gone into engineering. That's probably where my skill set lies. I think if I had had better math classes in high school, I probably would have pursued more quantitative career. I'm happy where I landed, of course, but I really hope we can find ways. And I think there's a lot of efforts in Maine, but we don't have, a huge research community. And so, there is this kind of, I think there's this migration of talent out of Maine, the kids that really show an aptitude and have a chance to go into research, often end up not coming back to Maine. They leave Maine and not come back. And it's because there really aren't that many opportunities.
I'm going to counter and argue with you a little bit.
Please.
I think the biggest issue we have, and I've discovered this through the festival, is that pure research and basic research, I agree with you, we don't have what other states have. And that's just a function of population. And it's a reason we're an EPSCoR state, et cetera, et cetera. But we have an extraordinary number of small organizations and small companies who people don't know about. And through no fault of their own, right? It's not their mission. Their mission is to do their work and to survive. So I think those of us who do know about the existence of these places, it's up to us to remind people that they're out there.
Yeah. And how do we do that? I mean, how do you get people to listen to a podcast like this, who wouldn't know it exists? Do you tell people that there are scientists out there that want to help you?
I will say the single most rewarding thing I've had with the festival, and, in the before times, before the pandemic, and that I hope, and in planning that we will go back to with 2022, is the fact that we can bring together over 10,000 people in the middle of March to talk about science, right? Like you got a sliver of it with the 5-Minute Genius, but the weekend where, it's the number of people who I overhear who come up to me and say, I had no idea that, and fill in the blank. And it is always that we had this in Maine. And it's not the kids. The kids just take it for granted if you get them there. It's the adults who just have no idea. I know it's there. Like, I know that all I need is if I can get one person who has no idea that we are around. And I don't mean to just focus on the festival. I think there's a lot of different ways to do it.
No, I think you're making a real difference.
But the way that we've approached it is come and learn or not. Doesn't matter. Just see what we have. One of our field trip days, the Maine Marine Sciences School brought in 2 fish to dissect during the field trip day. So throughout the day, there's this tuna and this shark on a table.
Oh, wow.
It was awesome. And as the day went on, they dissected the eye and showed the lens, and it was great. It was my only regret was we didn't do it with 5,000 people in the room. So I think we could do that. So it's really interesting because it becomes this whole, I can't believe they got a fish to cut up right over there.
Yeah. Yeah. I mean, one of the reasons I fell in love with biology in high school was I had really amazing teachers, really amazing science teachers. And, I didn't have a father in the picture at that time, or now for that matter, but I had amazing male role models, and most of them were my teachers. And my biology teacher, who was also my botany and zoology teacher, he I don't know where he got the resources. He must have spent some of his own money, but we would dissect. It seemed like we were dissecting, a new different kind of animal every week. And it was just, it's just incredible. You start to understand that all of these different organs, 500 cell types in your body or more, they all derive from one cell. The fact that one stem cell can produce all of these different cell types, and the choreography that happens between them, it's just it's amazing. Any of us are healthy because you think about how do 30 trillion cells in your body, one mutation can cause cancer that can kill you, and yet most of the time it doesn't happen. And it's it's amazing. I think showing the kids those really cool stories makes a big difference. I think there's a reason we send landers to Mars, and it's not just to see if there's life there. I think it's to really stimulate kids. I mean, my kids are amazed by that new Perseverance rover and what it went through to get there. And, we were watching the Nova special on PBS about it, and, showing all of these researchers and their stories, and, the choreography between thousands of people to get that lander over to a planet and down safely right in the spot where they wanted it. It's incredible. And I think that's worth it in itself. That's worth the investment in space, in research, because I just think does so much for kids and to stimulate, that interest in science. And I think we can do more of that, in basic science and in genetics.
So the two parts of that I think is really interesting. Number one, it reminds people that it's not just one scientist alone in a room.
Right.
There's that. Number two, total aside, did you know about the heat shield with that? Did they talk about that in the Nova? And it's a Maine company.
Is it really?
FMI Fiber Materials. They've been around 52 years. They built the heat shield for that and
ends
other
up
rovers.
Coming off.
Yeah. It's the largest heat shield ever made. They designed it and built it in Biddeford.
That's amazing.
They are an extraordinary company that do this tons of work.
So that's what you're talking about. There's lots of these.
That's what I'm talking about. These little. So I'm going to send you.
Science.
Yeah, I'm going to send you a little
I'd love to read about it.
And you can tell your kids they made it in Maine. Like
wow.
That heat shield is why Percy, I noticed on Twitter they're calling him Percy.
Percy
has their own Twitter feed. So I'm gonna make you focus back on what you do now. What do you do now?
Yeah,
it's not mouse cone ads, right?
Not anymore.
Yeah, okay.
But I am still interested in how cells make decisions to become different cells. And so I still work on stem cells that are making those decisions. And I still work on using genetic diversity, our genetic differences, as kind of a tool. And so, on a daily basis, I sit in my home office and talk to people on Zoom and WebEx. But in the before times, we would meet in person in the lab. And so, most of my lab is computational. We work on giant data sets out in the cloud. And essentially what we do is we get genome sequences for all of these genetically diverse mice. The reason we use mice is because you have a lot more access to tissues. We can study development. We can make mouse embryonic stem cell lines, which we can't do with humans. And so it's amazing how similar mice are to humans. We always think humans are so different, but really if I showed you a mouse heart, you probably wouldn't be able to tell the difference between a human heart if I was zoomed in far enough. So we have thousands of genome sequences for these individual mice or for individual embryonic stem cell lines. And then, we measure all kinds of traits for these different mice and lines.
Can you just explain, what do you mean exactly by a genome sequence?
Yeah. I mean, so, each of us has in every one of our cells, we have two copies of 2 billion nucleotides. And so, a string of A's, C's, T's, and G's, you get one copy from your mom, one from your dad. And any two of us, if we were to compare those strings, we would see that we differ at about 10 million of those individual nucleotides.
So that's what you mean is like the exact.
Exactly. So what I'm trying to find out is how do these mice or people or cells, how do they differ in their genome sequences? Because if I know where they differ, I can use that to find out, what is causing the differences later on in disease. So if disease has genetic causes, we can use this information to help us identify, okay, these people are more susceptible to disease, and they tend to have a specific base at this location in the genome. And so that can tell you that, there might be a gene nearby that base or a mutation that is actually causing, that disease susceptibility. So you're really, when I say I'm using genetic variation as a roadmap, I really am using it as a roadmap because I'm looking across the genome at these spots that differ between us, and then essentially correlating those differences with differences in phenotypes. And so I tend to focus on, what are the initial things that differences in the genome affect. And so normally, a genetic variant or a mutation is going to affect the expression, the amount of a gene that's made, or the function of a protein that a gene encodes. And so, those are what we call, proximal effects. And so there's these early steps of gene regulation that I'm most interested in that then ultimately lead to differences in cell fate decisions to differences in disease susceptibility. And so when I say we use genomics, we're essentially in every one of these animals or cell lines, we're counting the number of mRNA molecules that each gene is producing in each one of these individuals. And so by doing that, you get, let's say there's 25,000 protein coding genes in your genome. So you can tell the differences and you can link them to differences in the people's genomes or in the mice's genomes. And then you can start to measure different traits the further you get from the genome. So you can measure the amount of proteins, you can measure the amount of different cell types. You can slowly build that chain that starts with this genetic difference, and ultimately you can link that to differences in disease susceptibility. So it really is this, I tell my mom it's this, and I think I talked about this at the Maine Science Festival. I do these like multi-dimensional Sudokus. It's essentially, we're measuring, we have some numbers and we're trying to fill in the rest. We're trying to use the numbers we have to infer the numbers that, the traits that we're not measuring or that we're not able to measure, just as you would do like a Sudoku. But this Sudoku changes over time. And so, what is happening at one stage of development might be very different than what is happening when you're, an adult at the genome level. And so it changes over time, and it changes with each of these layers of traits that you're looking at. So if the genetic variation that's affecting the amount of mRNA for a gene might actually not be affecting the amount of protein that is made from that mRNA. And so there's all of these different codes that we're trying to kind of crack that link genetic differences to disease susceptibility differences.
It's exhausting to think about all the different iterations. And I'm assuming part of the reason why it's so unknown is because really this is such a new field. Is that true?
Yeah. I mean, this field that I work in is called systems genetics. And really it's combining, genetics, genetic mapping, using these roadmaps with these really high dimensional phenotyping, or, being able to measure what the genome is actually producing. So, you're looking across 25,000 traits across 10 million of these variants in the genome. It ends up being these, huge regression analyses to be able to figure out what are the genetic variables that are actually affecting that end variable of disease. So it is a lot of statistics. I'm not a great statistician, but most scientists do like the same set of things every day. So if you get good at the same set of things, you can just apply them in different ways.
Is there a specific disease that you're looking at, or is it not that? Is there just a broad overview or part of the gene that you're looking at specifically? Yeah,
I'm not obsessed with a single disease or a single gene. A lot of scientists will spend their whole career trying to understand a single gene. I'm more interested in using this type of way of doing research and applying it to really interesting genetic puzzles. And so a lot of my focus is understanding stem cells and how this genetic variation affects the ability of some of these stem cells to produce different cell types. I think, those are really fundamental questions that can be applied to multiple diseases. I'm starting to try to apply this type of genetic approach to cancer research. So, we have a huge cancer center at the Jackson Lab. I'm interested in kind of being a part of that. And so, my lab, people in my lab, we all work on a lot of different projects, but the common thread is this way of doing genetics. And so, when we meet, we still speak the same language, but someone might be working on Cornelia de Lange syndrome, or someone might be working on this, stem cell project, but we're doing a lot of the same things.
So it's a lot of what I would call, and I think it's still called, this basic foundational research that can then be built off of to figure out the more specific stuff, whether you're doing that or not. Yes.
Yeah, absolutely. Yeah. And, one of the things we're trying to do more of is to understand how your genome interacts with your environment and how that affects things, so adding on environmental diversity into this equation really changes things.
So the multi-dimensional Sudoku wasn't complicated enough. You needed to add in the environmental aspect as well.
Yeah, no, it's more interesting to do it that way. I mean, people don't live in a petri dish, right? And so in the lab, we can try to isolate the environment as a variable so that we can look at the genetic variable. But just like, genes don't act as islands, genes interact, and genes also interact with the environment. And so if you're exposed to PFOA or something in your drinking water and someone next door is not, your genes are going to be acting differently, based on how they interact with the different chemicals and things that they're exposed to. And so it's something I don't want to just identify things that might only apply in a petri dish or in a mouse cage. I want to understand, how can these genes, what happens if you introduce variation in the environment that they find themselves in? And I think that makes it more applicable to human disease research.
It's a really good reminder that our genes aren't this standalone, once it's set in your body, it's set. Right.
Yeah, right.
There's different triggers. It's not, I think for me, it's easy to think, DNA, genes, they're there, whatever. But different things are getting triggered all the time, right?
Yeah, it's, you have to be comfortable with uncertainty and probability. And humans naturally are not. We are binary thinkers. We want the light to be on or off, right? But that's not how biology works. And that's not how your genome works, so at the end of the day, if this stem cell is going to decide to become one cell or another, often depends on, what is the sum of all of these complex interactions between different genes and proteins in that cell. And then, there's still a probability that it could become something else, but, you're essentially, those networks shift probabilities. And so it's, if you're comfortable with uncertainty, then adding in other variables like environment is, it's fun.
If this last year has taught us anything, it's that uncertainty and probability are a really difficult concept to wrap your head around and to wrestle with on a daily basis for an awful lot of people.
Yeah, I agree. And I felt that too.
Yeah.
And that's the funny thing is I've meandered in life. But in the last year, I've really come to love routine. And that's helped get me through, a lot of this is knowing that every morning, I'm going to wake up, I'm going to pet the dog, I'm going to take a shower, I'm going to make coffee a specific way. It's having that routine kind of adds a sense of normalcy when everything outside of, your door is completely different. Yeah, it's been a really difficult year for everyone. And that's another way that the guilt comes in, is because I've been able to work from home and to continue doing what I'm doing and getting paid for it, and I see a lot of people back home who have lost jobs, who have really been adversely affected financially and who've lost loved ones, from COVID. I look forward to getting 2 shots in the arm as soon as possible.
Steve, this has been a delight.
Oh, I've enjoyed it.
I really appreciate it. The Maine Science Podcast has received support from the Maine Technology Institute and is recorded at Discovery Studios at the Maine Discovery Museum in Bangor, Maine. The Maine Science Podcast is produced and edited by me, Kate Dickerson. I received production support from Maranda Bouchard. The Discover Maine theme was composed and performed by Nick Parker.